Hello, everyone. My name is Ed Vital. I'm from the University of Leeds, and I'm chair of the Lupus Forum. Welcome to this special Lupus Forum webinar, which is about the challenges of clinical trials. This is aimed at those of us who are conducting trials at hospitals, but also perhaps some of us who might be involved in the design and running of these trials, whether in academia or in industry. I think many of us are familiar with the problems, and the idea is today to discuss some of the solutions. And to do that, we're very privileged to have three of the most experienced lupus clinical trialists you could ask for in this field: Professor Richard Furie, Vibeke Strand, and Professor Brad Rovin. So I'd really encourage you to take the opportunity to learn as much as you can from their experience while they're all here together today. We're going to start off with me giving an introduction to some of the main themes, and then I'll ask our speakers to take a deeper dive into these subjects. You can ask questions at any time you'd like in the Q&A box, and at the end of all the talks, we're going to go through as many of those as we can, given that time. So we've got a lot to do, so let's get started. So this is just an introduction to our faculty. I'm sure many of them are known to you, and I'll give some more detailed introductions as we go through. Here's the Q&A box that you need to look for. So if you just look at that Q&A box, put them in there, and we'll be able to go through those. So on to the learning objectives for this session. So the learning objectives are to understand the key challenges of conducting clinical trials across lupus and share different perspectives on strategies to address these challenges. So we're going to look at the key challenges in both non-renal lupus and lupus nephritis, and look at potential solutions and also what resources are available. And here's the agenda. I'll be giving the overview of the key challenges, and then we'll go to talks on increasing patient numbers and diversity with Professor Furie, heterogeneity and outcomes in trials with Professor Strand, and optimizing trial design and outcome measures in lupus nephritis. And then at the end, we'll all come together to discuss the resources and the questions. So on to a brief overview of what I think are the key challenges. You may disagree, of course, and feel free to add your own views when we get to the questions. So I think we're seeing a significant increase in the number of lupus trials, which is due to advances in the pipeline of treatments coming through to be assessed. I think with that increase in numbers, there's more limited awareness of the ongoing trials, and that is impacting recruitment. That's especially difficult because trials tend to be conducted only in certain specialist centres. I think that there can be a lack of diversity in the clinical trial population. That could be demographic diversity due to something like the ancestry of patient groups, or it could be due to the disease features. So there's an issue with disease heterogeneity. There's also issues in getting these trials to succeed, as in to show us meaningful and measurable outcomes. And from the point of view of the sponsor, there are some major challenges in the field in things like regulatory and logistical challenges. So this is a figure from a review article published last year by Laurent Arnaud's group, which I quite like as a snapshot, already changed, of course, but a snapshot at the time of the clinical trials landscape. So here we see all the different therapeutic targets or molecules being considered that are in early phases or in phase 2, phase 3, or actually reaching the market, for which only 2 were listed there. So there's a great drop-off as we go down towards marketing, but also what you see here is that these are in extremely diverse mechanisms of action, whether that is B cells or intracellular signaling or complement targeting. So the landscape is difficult, and essentially there are too many drugs and too many trials for us to be able to assess if we don't improve how we do it. And this is a natural thing to happen because lupus immunology is complex. So of course there are many therapeutic targets that make sense, but that means there's a lot of work to do to assess them all. This isn't helped by the fact that lupus is quite an uncommon disease, so it can be quite difficult to navigate this landscape. Which are the right trials to open? and if you have several trials available, which are the right ones to allocate particular patients to. Another problem is the success of the trials. So lupus trials don't always tell the truth. And I think we can see that with this summary of some of the more major trial programs that have occurred in recent years. So there's the BLISS, two phase 3 trials for belimumab, Embody, 2 phase 3 trials for eprotuzumab, TULIP, 2 phase 3 trials for anifrolumab, and the BRAVE, 2 phase 3 trials for baricitinib. So a few things to highlight here. One is that the standard of care response rate is high, commonly, not uncommonly, over 40%. That could be due to active background therapy. There might also be regression to the mean issues in that this is a naturally fluctuating disease and we recruit people when they flare. Similarly, related to that, the deltas appear to be quite small. So even though actually when a patient receives the right lupus treatment in the clinic, it can be quite impressive to us, it's quite difficult to demonstrate those benefits in clinical trials, even for drugs that I think are really good. And then my last point is that some trials, the reason I say that trials don't always tell the truth is that you can have two almost identical protocols of the same drug where one's positive and one's negative. So they can't both be right. And some of that, not all of it, but some of it relates to outcome measures, which we're going to talk about quite a bit today. Why are outcome measures difficult in lupus? Actually, I think a lot of people get frustrated with those of us who created the outcome measures, but actually it's not just that the outcome measures were badly designed. Maybe they could be improved, but actually they're quite good. The problem is lupus is just a difficult disease. You've got inflammation in every possible organ, and that means that there are so many other different diseases that need to be excluded when you assess each organ. We don't want to score eczema as a rash, for example. We have to make sure we're scoring only lupus. So there are strict rules for each feature. So there's a lot of organ features to go through. Each one has strict rules. But unlike other rheumatic diseases, there's this decision about attribution. In rheumatoid arthritis, if you squeeze a joint and it hurts, you put tender. You don't think about whether it's due to the rheumatoid or something else. If it hurts, you put tender. That's not how it works in lupus. In lupus, we ask you to only score things that are due to the lupus and due to disease activity. And that means that requires thought and experience in lupus to know what you're doing. You're making a diagnosis every time you tick a box. All of this needs training, and as in when you take a trial, you'll be asked to undergo some training, and all of this needs adjudication, as in behind the scenes, what you enter is being checked. And lastly, just to introduce this concept about the diversity, this is a really crucial issue in lupus because the groups of patients who are most commonly affected and most severely affected by lupus differ. Usually within each country, there'll be different groups of patients who are more affected or less. And some of the groups who are more affected may be those who are harder to engage in trials, like patients who are younger, or patients who maybe are from immigrant populations and speak a different language or have different illness beliefs. It's an important issue in access of the right people to the right trials. We also need to know how to code these differences, which can be quite problematic. For example, if we describe a group of people as Asian, that can encompass some very different groups of individuals. And also, as I mentioned, these trials are in an uncommon disease. They're also quite difficult trials to do sometimes. So that always means they're going to be happening mostly in specialist centers. And that means that a lot of lupus patients or physicians who aren't in those particular specialist centers wouldn't have access to trials, even though they might have patients under their care who would be very suitable for a trial and might derive great benefit from it. But just to finish up, I've told you about the challenges, but I think the reason we're all here to talk today is because we all also recognize it's a time of great opportunities. So having lots of new medicines is a good problem to have. That's what we need in lupus, and we can make the most of that opportunity. Also, although some trials have failed when people felt that they should have succeeded, that problem is getting better. We're getting better at doing them over time. And there are a lot of resources and also a lot of innovations that are going to improve the trials in the future. So at that point, I'd like to hand over to the first of our guest speakers. So Professor Rich Furie is going to present on increasing patient numbers and diversity in SLE trials. So for those who don't already know him, Richard Furie is Chief of the Division of Rheumatology at the Department of Medicine in New York and Professor of Medicine at the Zucker School of Medicine, where he's also working in the Department of Molecular Medicine. He's director of the SLE and Autoimmune Disease Treatment Center and of Northwell Health's clinical research program in MSK disease, the program in novel therapeutics. He's served on many committees of the ACR, including the board of directors, and you really couldn't ask for anybody more experienced in lupus trials. So it gives me great pleasure to hand over to him.
All right, thanks, Ed. Here are some disclosures. All right, so in the next 15 minutes, I will cover these 4 topics. Let's start out with the epidemiology of SLE and relate it to trial enrollment. In fact, let's go back about 20 years. The belimumab phase 2 study in SLE had failed, but a post hoc analysis eliminating from the analysis patients who were serologically inactive yielded some favorable signals. And so the estimated effect size from that post hoc analysis in the phase 2 study was somewhere in the teens. And there were enough signals to move forward into phase 3. So what did the statistics predict? Well, based upon an effect size, an estimated effect size of about 14%, with a 5% significance level and 90% power, it was determined that about 270 patients per arm would be needed. With 3 arms, placebo and 2 treatment arms, 810 patients were needed. And of course, to get regulatory approval, 2 studies were needed. So we're talking about 1,600 patients for the phase 3 belimumab program, the so-called BLISS program. Well, where do those patients come from? There are a lot of numbers on this slide. These are epidemiology studies that were recently done in lupus. And lupus falls somewhere between rheumatoid arthritis and scleroderma as far as incidence and prevalence. But the most important line is the one about 5 or 6 down, where I have highlighted 204,000 patients in the United States could be classified as having lupus. And this was published just a couple of years ago. That's not a lot of people with lupus. And in fact, you figure about a 1-to-1 ratio of SLE to lupus nephritis, then you're dealing with about 100,000 people in the United States who have SLE and are potentially eligible for an SLE trial. Now, we have an additional problem that Ed alluded to, and that is diversity. So this is from the same study by Pete Ismerli, and you see a ratio in females of 2-to-1 whites to blacks. Is that representative? Or is that represented in our enrollment in clinical trials? Absolutely not. We have an underrepresentation of blacks in our lupus trials. Now, it may be a little bit unfair. I'm showing you the TULIP-2, the anifrolumab trial, and you can see about 4 or 5 lines down that there's roughly about 10 to 13% of enrolled patients who are of African ancestry. But these are global studies. But at best, I can recall the phase 2 belimumab trial, which was done largely in North America, where about 25% of the enrolled patients were of African ancestry. So we have a diversity issue with enrollment. All right, let's move on to the ideal clinical trial participant. Now, it's very important. Classification criteria are not diagnostic criteria. So the patient may think they have lupus, the clinician may think the patient has lupus, but they have to meet classification criteria of some sort. There have been several iterations of classification criteria over the years. They have to meet these criteria to be eligible for the study. And here are the latest classification criteria. These are the EULAR-ACR criteria. I'm not going to go through all of these items, but in order to make it classifiable as lupus, one at least has to have an ANA that's positive. And then you can see on the right-hand side, one needs to accumulate points, 10 points, to be classified as lupus. So that's hurdle number one for a clinical trial. Hurdle number two for a clinical trial is having enough disease activity. And I'm showing you SLEDAI 4-point categories. Generally, for our SLE trials, one has to either have a certain amount of BILAG activity or a certain amount of SLEDAI activity. And for SLEDAI, it's usually about 6 to 8 points at a minimum. And you can see for arthritis, that's worth a lot of points. That's worth 4 points. And in order to achieve those points, one just has to have 2 joints with pain and signs of inflammation. And unfortunately, pain and signs of inflammation could be or is in the eye of the beholder. And then you see some of the other values worth 4 points. All right, here are some of the 2-point items for SLEDAI. Rash. Mild rash could get you in. Hair loss could get you in. So now we have 2 points for rash, 2 points for alopecia, and throw in mucosal ulcers, some oral ulcers, and you're up to 6 points, which may make someone eligible for a study. This could represent very mild lupus that could respond very easily to placebo. So we have a big problem. This has been our nemesis for decades, the high placebo response. And in fact, here's a good example, a recent example. This was a low-dose interleukin-2 study. And you can see the placebo response in blue approaching 60%. You can't show a drug is effective if your placebo response rate is 60%. Now let's also go back in time to the phase 3 belimumab program. So the question is, could we do something to identify patients who might be better responders, who would have a high rate of response? And so that led to the phrase high disease activity, HDA. And so in this particular analysis, this was a postdoc analysis, first author was Ronald van Vollenhoven, where the BLISS-52 and the BLISS-76 data were combined. There was a multivariate analysis, and then the results of that analysis were transformed into items that we kind of do every day on our patients, namely check serologies. So if one looked at those who were DNA antibody positive at baseline in the BLISS studies and those also who had low complement, that constituted a high disease activity patient. And I'll just show you data of how these high disease activity patients performed in the study. And the high disease activity group represented about 50% of the original cohort. So here we go. This is the native cohort showing you response rates using the SRI as the outcome measure. In, maybe hard to see the colors, but in blue, you have those who received belimumab, and in gray, those who were in placebo. And remember, in our studies, placebo is not just placebo. The patients are coming in on background therapy, which typically consists of antimalarial, steroids, and immunosuppressives. Maybe not 100% are on all three, but roughly about 3/4 are on background steroids, about 2/3 on background antimalarials, and about 50% on background immunosuppressives. So you can look at the effect size on the right-hand side. It comes out to what, about 11 percentage points or so. Then if we look at the high disease activity patients, representing about 50% of the original cohort, you see that the response rates for those who receive belimumab are about the same as the native cohort. But interestingly, if you look at the placebo group, in those who had high disease activity, the placebo response rate is lower. So the effect size grew to about 20 percentage points. So what did this mean for future SLE trials? Every sponsor wanted high disease activity patients, SLEDAI of 6 or sometimes SLEDAI of 8. Where are all these patients coming from? All right, let's talk a little bit about trial design challenges and implementation. So as I just mentioned, there are inadequate numbers of patients and there are inadequate numbers of informative patients if one wants those SLEDAI patients of 6 or 8 or greater. Now, there's a lot of criticism about the endpoints not being ideal. The SRI depends upon the SLEDAI. The SLEDAI was not invented for clinical trials. Then we have the BICLA, which is the major driver of efficacy is the BILAG. So there are groups working on better endpoints. I mentioned background therapies, and there's no question that the background therapies that the patients are taking at entry are confounding. We try to train investigators, but when these studies become global and involve hundreds of sites, investigator training may not be optimal. And as Ed said, there's intense competition right now. In fact, I just got back from EULAR. Someone said there are about 29 companies pursuing CAR-T in lupus alone. There are many companies pursuing TYK2 inhibition. There are many pursuing inhibition of the interferon pathway. So we've never seen this before. If you go back to the 1990s when we first started studying these novel therapies for lupus, there were just 2 companies. And as I mentioned, the placebo response rate has been our major nemesis for the last 25 years. So how can we lower the placebo response? Well, we can try to enroll informative patients, very active patients. So besides using the SLEDAI, maybe we could use a biomarker, but that biomarker has been quite elusive. Not so much in SLE, but maybe Brad will talk about weeding out placebo responders early on. That's a design that we've used for a couple of lupus nephritis trials, meaning there's a run-in period, and those who respond to standard of care are no longer eligible to be randomized into the trial. That's only been done in 2 trials, and I'm not sure that it really worked. We could use more rigorous endpoints, such as remission or LL-DAS. So the discrimination may be greater even though the response rate's lower. One design is to withdraw background steroids or immunosuppressants, and that's been done in a few studies, kind of the sink or swim approach. I'm not a big fan of that approach, but it's out there. And again, site selection, very important, training, very important. And we've had a problem where phase 2 is not replicated in phase 3. And why is that? Well, there's a larger enrollment. We have to go worldwide. Broader geographic involvement, obviously. The site training may not be as effective. And perhaps the adjudication not as rigorous. And we've learned that adjudication can help, but it needs to be quite rigorous. And I'll just end with examining the current trial landscape, and that's easy. There's one slide to show. Right now, there are more trials than there are patients. I've been saying that for several years, and we need to expand our trial designs because there are needs of lupus patients beyond the high disease activity patients. There are patients who need to get off steroids, patients who need to get off their immunosuppressants. We need to prevent damage. And I could go on and on and on. But it's tricky designing trials to accommodate these other needs. You have to get regulatory buy-in, and you have to get sponsor buy-in as well. But we've come a long way since the early 1990s when we were just studying 2 drugs. And we've had 3 drugs approved now for 4 indications. And no doubt there will be more drugs approved in the near future, and that just means better outcomes for our patients. So thanks, Ed.
Thank you very much for that extremely comprehensive overview. So I'm sure there'll be lots of questions, which we'll come to at the end. But before we do that, I'd like to hand over to our second speaker, which is Dr. Vibeke Strand, who's an adjunct clinical professor in the Division of Immunology and Rheumatology at Stanford University School of Medicine. Since 1991, she's led a consulting practice offering clinical research and regulatory strategy expertise to pharmaceutical and biotech companies for developing new therapies in rheumatic diseases. She's participated in the development of all the approved biologic drugs and targeted synthetic DMARDs in RA, as well as new therapies for PSA, SPA, SLE, vasculitis, gout, biosimilars, symptomatic therapies for osteoarthritis, and many more, with over 500 publications. She's a fellow of the American College of Physicians and master of the American College of Rheumatology. Professor Strand has played a crucial role in helping to systematically organize all these complex outcome measures, as well as patient-reported outcome measures that we use in lupus, and that's what's made trials successful. So over to you, Vibeke, to take us through this difficult topic.
Thank you, Ed. So these are just my disclosures. Well, as previously said, it's challenging to assess outcomes in lupus trials. It's obviously a multi-system heterogeneous disease characterized by flares and remissions. And when we're trying to do multinational trials, which we have to based on the size of the population, the patient population, we have to account for differences in medical practices, as well as all the other heterogeneity that comes up with doing worldwide trials. There's genetic predilections for more severe progressive disease, often in minority populations as African Americans, Hispanics, and Asians. We talked about enrolling a responsive patient population based on the high disease activity subset of the belimumab trials, but those patients are not as common as we would like them to be. And we do have other needs of the patients. And identical trials run in parallel to phase 3s that are essentially very similar in design often don't yield replicate results. And it would be better if we went to 2 different designs. So we talked about the challenges of assessing outcomes in lupus. There's really limited regulatory precedent for successful approval since we only have 3 so far, and we are expecting at least 1 more in the near future. So when does standard of care as background treatment become unethical? And could we do an active comparator trial and therefore not have a very prolonged exposure to placebo? So far, that has not been done. Obviously, ongoing data review and adjudication is very important. And that is also because using either the BICLA or the SRI-4 is not simple. Neither the SLEDI nor the BILAG were designed for trial use, and they've been adapted as such, but that's just another reason for adjudication to be important. And a reason for definitions of treatment failure are essential, and that they be met during the trial for patients who would be considered non-responders. Allowing changes in background standard of care does confound results, and yet we know that tapering of glucocorticoids is essential and now becoming part of the primary outcome measurement in several trials. There's a lack of consensus regarding the responder indices, and they're not necessarily considered clinically meaningful. Definitions of low disease activity, such as the LL-DAS and DORIS remission, are more clinically meaningful, but they haven't been used as regulatory outcomes except in the secondary outcome list. Responder indices should be supported by reductions in glucocorticoid use and by clinically meaningful improvements in patient-reported outcomes, which are quite important in lupus. Fatigue is a major component of the impact of lupus disease activity, so as is health-related quality of life. And we have both disease-specific and generic measures for health-related quality of life. Coming back to the selection of patient populations and remembering the BLISS trials. Yes, that's an example of why we need large sample sizes, which also makes it difficult in a disease such as lupus. If you look, not only the belimumab trials had 1,600 patients, tabalumab trials had 2,200 trays, and Embody 1 and 2 with eprotuzumab had 1,500 patients in total from both of those trials. We don't have homogeneous patient populations in lupus. However, you would like to subset the patients. We've seen in the ALMS trial that mycophenolate mofetil was more effective in African American and Hispanic patients than IV Cytoxan, yet the trial overall failed. In Explorer, rituximab was actually more effective in African Americans and Hispanics, yet again, the trial failed. In BLISS 76, belimumab appeared to be less effective in African Americans, but was more effective in lupus nephritis and in their phase 2 population. So it's also not clear that any certain agents are more effective in specific organ system disease, yet we must actually assess that specifically with every outcome in a trial. We have to remember patients with lupus nephritis also have extrarenal disease, and it's important to assess it. And typically subjects with significant CNS and renal manifestations will be excluded from trials because of the fear that they will deteriorate and the fact that we can't assess CNS disease in a trial-worthy manner. Just to go back to the high disease activity argument, here are the trials with belimumab, anifrolumab, tabalumab, atacicept, the IL-6 from Pfizer, ustekinumab, and baricitinib. And we can see that those patients who were double-stranded DNA positive with low complement levels, C3 and or C4, those were the patients who tended to have more active disease. They were the enriched populations, and they were associated with improved response rates. But again, difficult to find all these patients to accrue into large trials. Coming back to outcome measures, none of them were designed to be used in trials. So we're thinking about both the BILAG and the Selena SLEDAI or the SLEDAI 2K. Some allot new points for manifestations that have just occurred, but no points for stable disease. They have different horizons for comparison in terms of timing. The organ specific is really more BILAG than SLEDAI, but the SLEDAI is weighted. Only the BILAG and the SLEDAI 2K RI50 will record partial improvement. It's also important to assess moderate and severe flares over time. Yet we oftentimes have a flare index that combines both moderate and severe, yet we want to separate those. We have the BILAG with A or 2B flares. We have the International Flare Index. And then we're looking at the evaluations in these trials. Must we adjudicate them formally, each evaluation? Because that is a lot of work, but it is necessary because there's too much variability in assessment. And part of that has to do with the experience of the investigator. How often have they used the BILAG as well as the SLEDAI? And how experienced really are they with them to know what the problems can be in terms of needing adjudication? So the responder indices we now have required validation in one trial and confirmation in a second trial. And that's now occurred both with the SRI-4 and the BICLA. Just to remind you about the SLEDAI, we've talked about it already, but there's 24 components, yet the scores rarely go above about 12 to 15. The presence or absence of manifestations, but not the severity, is what's scored. And we do like the SLEDAI 2K RI50, which will score 50% improvement, but that's not necessarily what's used. These are weighted manifestations, but notice that they're very different according to the organ system. And as I said before, rarely patients have scores above 15 or 20. And yet we think severe disease is essentially 10. We talked about the dsDNA antibodies, low complements, leukopenia, and thrombocytopenia. These are the only blood determinations scored in the SLEDAI. If they're all abnormal, they will account for a total of 6 points. Yet we've seen other organ manifestations that are certainly at 4. And the SLEDAI doesn't include pulmonary hypertension, hemolytic anemia, TTP, mesenteric vasculitis, or pulmonary hemorrhage. And we do agree that the clinically meaningful improvement is 4 points or above. The BILAG is more complicated and is scored as an intention to treat, with each system graded from A to E. Flares, as I said before, are 1A or 2B scores, and oftentimes that's what's used for eligibility. And essentially, you're looking at improved, same, worse, or new manifestations. And then our responder indices. There's either the SRI-4 or the BICLA, yet they are very similar. First, the patient remains a responder. So that means no early exit from the protocol due to serious adverse event and adverse event intolerance or other reasons. And it means no addition of new medications. Although we do allow in trials up to 7 to 14 days of an increase in glucocorticoids of a certain limit. Second, clinically meaningful improvement from baseline to endpoint must occur. And with the SRI-4, that's an improvement in SLEDAI of 4 points. And for the BICLA, it's a loss of a BILAG A or simultaneously 2 Bs between baseline and endpoint. Third, there must be no worsening, no deterioration between baseline and endpoint. So that for the SRI-4, it means that the BILAG's included for no new BILAG A's or simultaneous 2 Bs. And for the BICLA, it means that the SLEDAI is included for no worsening between baseline and endpoint. And finally, there must be no worsening or deterioration in the physician global assessment, which is an increase in 10 points on a VAS or in the more commonly used Selena Flare Index 0 to 3 scale of greater than 0.3. So when compared head-to-head in phase 3 trials, interestingly enough, patients who are defined as responders by SRI-4 are not necessarily responders by BICLA and vice versa, which leads to more heterogeneity in our outcomes. Just to show you the difference between clinically meaningful low disease activity or DORIS remission, and then what we see with an SRI-6 or low disease activity. This is from the atacicept ADDRESS trial. And yes, it's a few years ago, but it was a very promising product. You can see the SRI-6 had about 52.5% responders. LDA dropped that to 26.6%. Then the LLDAS, which is a SLEDAI 2K of less than or equal to 4, without major organ activity or no disease new activity. MD global of less than 1, prednisone equivalent of less than or equal to 7.5, and stable immunosuppressives took it to 19%. And then the DORIS remission definition, which was the SLEDAI 2K of 0 MD global of less than 0.5, prednisone, prednisolone equivalent of less than or equal to 5 milligrams daily, and stable immunosuppressive or biologics. Now we have a remission rate of just under 11%. And protocol design and conduct is another challenge for us. You know, we could use one of our now 3 approved products for lupus, but we haven't had a non-inferiority comparison trial for labeling. And so we're still using new therapy versus placebo, which includes standard of care. That accounts for the highly variable placebo responses. It accounts for the need for definitions of treatment failure. And it also means that we have to consider no deterioration, in other words, stabilization and or improvement as an acceptable response. We've gotten increasingly stricter about use of background standard of care, and this is very, very important. That rescue or stress coverage with steroids shouldn't be longer than 7 to 14 days, and we should be including that preference for tapering glucocorticoids to less than or equal to 5 or 7.5 a day. There's where adjudication can come very importantly, and it would be nice to allow initiation of tapering of glucocorticoids as early as, say, week 4, and allowing individualized regimens can best differentiate active from background therapy. There really is no way to institute a generalized taper across a heterogeneous lupus population. So what do our patients tell us? These are the impacts of lupus: appearance, cognitive difficulties, fatigue, as we talked about, pain, unpredictability, emotional well-being is definitely impacted in physical function. And what do they want? They want improved psychological and emotional ways of thinking. They want better social family relations. They want better and less economic burden. They'd like to maintain their working life. Yes, they want treatment, but not so badly that it interferes with their life. Planning ahead is important and very difficult to do with a systemic disease like lupus. Daily activities are affected, and certainly body image. And so finally, now there are many efforts underway to develop better trial designs and outcome measures. And thankfully, hopefully this will go forward. So there's the Alpha Project, addressing lupus pillars for health advancement. There's treatment response measures in lupus, a tool effort led by Dr. Erin Brand at Monash University that was started by the Australia-Asia group. There's a FDA CEDAR has partners with Lupus Research Alliance to launch the Lupus Accelerating Breakthroughs Consortium, Lupus ABC. The Lupus Foundation of America has a rapid evaluation of activity in lupus measure developed with Drs. Joan Merrill and Anka Askenazi. And finally, outcome measures in rheumatology is an international effort to review and revise the recommended core outcome measure set for randomized controlled trials and longitudinal observational trials. And then this group will help to further validate new outcomes that are being developed by the above groups. So with that, thank you very much.
Thank you very much. Thank you very much for that. Again, extremely comprehensive overview. There's a lot of information to digest there, and I'm sure there'll be lots that people are going to want to discuss when we get onto the Q&A. But before we have time to do that, I have to introduce our third speaker. So our third speaker is Professor Brad Rovin, and he's going to present on optimizing trial design and outcome measures in lupus nephritis. Professor Rovin is the Lee A. Herbert Professor of Nephrology and the Medical Director of the Ohio State University Center for Clinical Research Management. He got his Bachelor of Science in Chemical Engineering from Northwestern University in Evanston, Illinois, and then his Doctor of Medicine from the University of Illinois Medical School in Chicago. And after his residency in internal medicine in Barnes Hospital in St. Louis and a fellowship in nephrology at Washington University School of Medicine, he joined the Ohio State University College Medicine faculty in 1990. He became director of the Division of Nephrology in 2004 and served as vice chairman of medicine of research from 2009 to 2019. I think that lupus nephritis has been amongst our most successful areas in lupus clinical therapeutics, with a number of licensed therapies that are fitting into a logical treatment algorithm and many more of those on the horizon. Brad's been absolutely at the forefront of what's gone on in this field over recent years. So again, I think we can learn an awful lot from him. Over to you, Brad.
Thanks so much, Ed. It's a pleasure to be here. All right, let me get going. Here we go. So I might have a little bit of an easier task because what I just heard was so complicated for a non-rheumatologist with all the different indices. It makes looking at endpoints and trial design for lupus nephritis straightforward because we have fairly objective measurements that we consider. So these are my conflicts. Okay, so if we think about all the clinical trials we're doing in lupus nephritis, the complete renal response is the endpoint that's used most of the time. I know that the BLISS-LN trial used a modified complete renal response, which was very similar with a little bit relaxed criteria. The complete renal response, basically, as you all probably know, is a combination of glomerular filtration rate or kidney function and proteinuria. Sometimes a urinalysis is included, and as was mentioned earlier, a glucocorticoid taper has been part of some of the responses in recent trials. The urinalysis, of course, if it's done by a knowledgeable person, can actually be quite helpful, but in trials that are done worldwide at many centers, some of them not having the appropriate access to microscopy or expertise, that can actually become a hindrance, and we've shown that in the past. One of the issues that comes up is that seldom do clinical trials look for a sustained complete renal response. In other words, looking at a single day in time in a dynamic disease with organ function that can fluctuate pretty dramatically, as well as proteinuria levels that can fluctuate dramatically day to day by dietary intake, fluid intake, salt intake, really begs the question of whether or not we should show this on a more sustained basis. Now, when you look at all the variables of the complete renal response, it turns out that proteinuria is the absolute most critical variable, and unfortunately, it's an unreliable marker. In new or early disease, lupus nephritis, proteinuria is a pretty good biomarker of lupus nephritis activity. However, as the kidney accumulates chronic damage, which really starts to occur at the very first episode of lupus nephritis, proteinuria can occur because of the chronic damage to the kidney or the scarring, and not necessarily from active disease. Sorting that out clinically becomes very difficult, and of course, histologically, we do repeat biopsies, but you can't do too many of them, as you all know. The other issue is that proteinuria due to chronic injury will not resolve with immunologic therapy, and therefore can be misleading in terms of a drug's effect. Serum creatinine, or GFR, is also a problematic variable, because what we're mostly looking at in complete renal response is a return to normal GFR, but we really don't know what the patient's true baseline is. Many of these patients are young and healthy before they got lupus, and they had no particular reason or interactions with the healthcare system to have had a GFR measured. And so achieving a normal serum creatinine or GFR is questionable. And if there's been damage to the kidney, you may never, ever achieve a normal GFR. So that becomes a very difficult bar to reach. So the other problem is CRR, complete renal response, is often differently defined for each clinical trial, making interpretation or comparisons between the trials quite difficult. So I listed what we usually use, a urine protein to creatinine ratio of less than 0.5 milligram per milligram of creatinine or less than 0.7 milligram per milligram of creatinine based on a 24-hour urine collection. That's often the criteria for CRR or the more recently adopted PERR that was used in the BLISS-LN trial. We like to see an eGFR greater than 60. I will tell you right now, my field has defined GFR greater than 60 as okay. I never want a GFR that's 61. I want a GFR that's much higher. So how this is really considered adequate is beyond me, or a GFR of 90 mil per minute per 1.73 meters squared. These are often the definitions that are used, more relaxed and more strict. Or more commonly, we see no decrease from baseline or pre-flare GFR of more than 10 or 20%. So clinical response is a little bit misleading, potentially. These are data that we have accumulated over many years in repeat biopsy studies, and I'm showing you our data, not to exclude anyone else's data, but other groups have shown similar results. These patients were biopsied at their very first presentation of kidney disease. They received mycophenolate or cyclophosphamide, and then had a protocol biopsy, no matter how they were doing, about 7 months later, generally when what we consider induction or initial therapy was completed. And what you see on the left is patients who had achieved a complete renal response with a definition of proteinuria less than 0.5 PCR. And you see a number of patients that have a very low histologic activity index, suggesting they had responded fairly well to the therapy. But you also see patients with activity indices above 4. And these included findings of endocapillary proliferation, subendothelial deposits, interstitial inflammation, and crescents. Again, showing that if you have a clinical complete renal response, you can still have activity going on in the kidney, and that can be very misleading. On the other hand, if you look at the right graph, patients who achieved a complete histologic remission after 6 months, some of them, in fact, a lot of them still had proteinuria greater than 500 milligrams a day. And so they would not have been recorded as a complete clinical response, again, skewing the results of a clinical trial. So we did similar studies after several years of therapy. These patients were induced in the usual way with cyclophosphamide or mycophenolate for about 6 months, and then they had 30 months of maintenance immunosuppression and at least 12 months of a clinical response. And then the biopsies were repeated on a protocol basis. And what you can see in the graph on the left is that only 44% of the complete renal responders actually had histologic remission, defined as no further activity, inflammatory activity in the kidney. And the other hand, you see that in the partial responders, which meant they had persistent proteinuria over 500 milligrams a day at the end of the treatment period, quite a few of them actually had an activity index of 0. So what does this all mean? Patients with a partial renal response could actually be in complete histologic remission, and they would not have been counted in a clinical trial, and could skew the results of a trial away from a good drug. On the other hand, patients with a complete renal response clinically could still have histologic activity and yet would be counted amongst complete responders, and yet followed over time in the future could have a poor outcome. So the question really comes up, could we or should we consider alternative endpoints for lupus nephritis clinical trials? And this is heresy. I'm being difficult, according to some folks, especially in pharma, because I'm changing something that the FDA has set up. I'm proposing to change something that the FDA has set up that has facilitated approval of at least 2 new drugs and probably a third on the way for lupus nephritis. And I think what we have to ask ourselves is, what are the real goals of lupus nephritis therapy? Well, I would tell you that the real goal of lupus nephritis therapy is not to get the proteinuria to a certain point at the end of 1 year of treatment. I mean, that's good. We should do that. Proteinuria often correlates with long-term kidney outcomes. But what we really want to see is preservation of long-term kidney function. And the way we do that is to slow the decline of GFR deterioration in a chronic kidney disease, and lupus is a chronic kidney disease. We also want to minimize the development of chronic kidney disease and or the tendency of progression, and we do that by reducing flares or preventing lupus nephritis flares. And this is just an implication of flares. For those of you, I think everyone on this call probably appreciates it. If you on the left panel, doubling of serum creatinine is the y-axis. It's prevention of doubling of serum creatinine. When you double your serum creatinine, that's a well-accepted hard endpoint in clinical trials of the kidney because those patients are on their way to end-stage kidney disease. And you can see that over time, you have some risk of this in patients that have their first flare, which is the top line. And then you see that more and more patients are going to double their serum creatinine if they've had more than one renal flare. On the right side is some data that we recently put out from a repeat biopsy study. And the worrisome part is that we did a biopsy at time 0 for the diagnosis. We did a biopsy at 9 months after initial therapy was completed. And again, these were not the new therapies. These are mycophenolate or cyclophosphamide. And then further on is a biopsy when the patients were doing really well and we wanted to see if they had no activity left to take off therapy. But what you see on the y-axis is the components of the chronicity index. And what's striking is that even during our very, very best therapies that we offer at the beginning of lupus nephritis, when we detect a flare, patients are accumulating chronic damage throughout that time period. And so that suggests that until we have a way to prevent fibrosis or scar accumulation, we really are only partially treating and preventing deterioration of kidney function and chronic kidney disease. So, are GFR and lupus nephritis flare trials feasible? And I would suggest to you that the results of contemporary clinical trials suggest they are. And the best example, or the first example really, is the effects of belimumab on kidney function. Remember, belimumab was unique at the time for BLISS-LN. It was a 2-year trial, and it allowed us to look at the change in slope of GFR over the 2-year period of time. And what you see in the first category is a negative 5.72. That means the loss of yearly GFR in the placebo group was about 5.7 mils per minute per 1.73 meters squared per year. Compare that to the loss in the belimumab group, which was significantly less. Okay, and what that implies is that we're preserving kidney function. Notice, it isn't 0, so we haven't made the decline in kidney function go away, but we're slowed it down substantially. And we calculate on a theoretical basis using modeling, saving 1 mil per minute per year of kidney function actually can extend the life of the kidneys, i.e. the patient does not need dialysis or kidney transplantation for several years. So this is really, really important. We also looked at harder endpoints like a fixed decline of 30% or 40% in GFR. Again, those were both favored in the patients receiving belimumab. And then if you look at the flares, these are flares after the first 6 months of the trial, you can see that there were significantly fewer flares in the patients receiving belimumab. Similarly, we saw that in patients in the obinutuzumab trial, which is now under a phase 3 trial, which is finishing up. These are data from the a sub-analysis of the phase 2 trial. You can see that the GFR did much better in the patients on belimumab, which is the dark line here, and this is the placebo group. And you can see that the slope estimate for GFR decline in the OB group was very little, 0.43, compared to about 4.5 mils per minute per 1 meter squared. Now, we have to confirm all that, and these numbers are smallish, but you get the idea that at least some of these B-cell targeted therapies are doing a better job at preserving GFR than what we see with standard of care therapy, and that's really critical to maintaining the survival of the kidney for the patient's lifetime. We also looked at 30% and 40% declines in the obinutuzumab trial, and that was actually similar to what we saw in the BLISS-LN trial. And this last slide here is also from a post-hoc analysis of the phase 2, and you can see in the bottom line there were significantly fewer instances of lupus nephritis flare in the patients receiving obinutuzumab than the placebo group. So, how can we put this together? This is my last slide before questions. I suggest that we really can start to think about a novel study design, and this is gaining traction for other glomerular diseases. And we've implemented this for IgA nephropathy, which is another immune complex mediated glomerular disease. We do a double-blind treatment, placebo versus the experimental therapeutic for 9 months to a year. And in the IgA trials, we can achieve accelerated approval for the drug based on proteinuria. In lupus, I would suggest we would base an accelerated approval on the CRR rate, and then we have a blinded follow-up period for another year in which full approval of the drug is based on a significant improvement in the decline of GFR over time, or GFR slope. This is feasible. We've had accelerated approval of these two drugs for IgA nephropathy. This was based on significant work with the nephrology community in conjunction with the FDA, and this is a different FDA subsection than what handles lupus nephritis. It has not been applied to lupus nephritis before. So I think what we need to do is start thinking about what's important to the patient over time, as was just Vibeke just showed us. And I think in lupus nephritis, what's really important to the patient is that they avoid dialysis or the need for kidney transplantation. And I'll stop here. Thank you.
That's great. Thank you very much, Brad. And I can already think of lots of questions from listening to that. So and we'll come to those in a moment. But we've got one other thing that we're going to go through, I think, as a group, first of all, which is just to come on to discussing from some of these theoretical problems with clinical trials and how we can design them and get to be successful, but to the more pragmatic issues, perhaps those facing a doctor who might be wanting to take part in clinical trials or to refer patients to trials. So we're going to have a look at some of the resources that are available that could be used. Okay, and this is an opportunity for everyone to comment, really. So I'll just sort of see who wants to say something, but I think feel free for all the panel to chip in with any comments here. So this is some of the areas of clinical trial resources that we thought about. There might be others. And if I just start on the top left, I talked in my talk and also in Richard's talk about how many trials there are and how we can navigate the landscape. And one obvious thing there is, are there databases that help us to do that, like to identify the trials available. There's clinicaltrials.gov, of course, which I find quite useful as a researcher, but I don't know if it's that helpful necessarily for somebody who's just trying to enroll a patient or trying to decide which trial to open at their site. I wonder, does anyone have any thoughts about this?
It's really hard to navigate, I think, for a person who's not doing this a lot. And I do think that I think through the Lupus Foundation of America, for example, patients can call there and get a resource of what clinical trials might be available in their area.
Yeah.
And I think the Lucin community might also be able to provide that information, both of which are really patient-forward organizations.
Yeah.
That's right. Yeah, I was going to say that. So the LFA website has a kind of list of trials open where you can find trials of different populations and the patients they'd be suitable for, where the sites are. And there's something, if you're in Europe, there's something equivalent on the Lupus Europe website as well. Again, you can find a trial and where it might be available. The other one, of course, being actually if you know a specific molecule of interest, is to look on the company's own website, where they'll often list that kind of information. Although I do think we could probably do a little bit better on developing those databases, and that's something we're trying to do in the UK, to kind of develop a site where doctors can identify the nearest hospital to them with a trial that would be suitable for their patient.
One of the things we're doing with lupus nephritis, a lot of kidney biopsies, at least in the US, are funneled into specialty or organizations that have a nephropathologist. We get a lot of biopsies outside of our university, and part of the issue is the nephrologist, review with the nephropathologist. And when we see lupus patients being diagnosed, we have the nephropathologist communicate with the referring physician that there are lupus nephritis trials open. Would they be interested in having their patient see us for one of those trials? And then we facilitate, coming to our lupus clinic. So that sort of thing is a little bit at the ground level, but it could be quite effective because there's major centers throughout the United States that see lots of kidney biopsies.
Exactly what I do. I know it's simple, but even just at our regional meeting, telling people the trials I've got open, or sending around emails to my colleagues sometimes and just saying, these are the trials, you may not just think about them. And I think maybe from the sponsor's point of view, they can sometimes facilitate that a little bit by providing you with like leaflets and posters and stuff like that you can send to people, or these little brief summaries of the key eligibility criteria, things like that.
Yeah, one just has to be very careful about patient privacy.
Yeah.
No question. And that's why we have the pathologist tell the referring doctor that is a possibility, and then they can contact us. We never go straight to the patient.
Yeah. Okay, and so actually maybe at that point we could talk, are there other things that things like the patient charities, research alliances, patient advocate groups offer that are important here, apart from just finding trials? Supporting patients who are considering a trial, perhaps?
Well, Brad and I were on a committee of the Lupus Foundation of America. How many years ago was that? Probably about 7 or 8, where they had received a grant from the government. And they were trying to figure out how to get more African Americans involved in clinical trials. And that was to be accomplished through the Black Churches of America. But I think by the time it came to implement the strategy, the grant ran out. So I don't know whatever became of that.
Yeah, it's unfortunate, but we actually, in our local community, sometimes do health fairs at community centers to sort of say, hey, there are these things out there that are available. But Rich is right. We had momentum and then we didn't.
But I sometimes think something I've found is that the from the point of view of a doctor who works in lupus, I don't know everything that goes on in the patient groups, you know, like the forums and Facebook groups and things like that. But there's an awful lot there that can be very helpful to patients. And people with lupus often don't know anyone else with lupus. And when you say something like, do you want to take part in a clinical trial, that may sound quite intimidating, it can be quite helpful to just simply contact someone else who's been in a lupus trial before, and or find out what they think about it or what questions they should ask.
Mhm.
Well, but there's a lot of online groups now
Yeah.
that are not just arthritis, but are lupus. And I think it's quite helpful. I know when I was in practice, when I would go into round on Saturday mornings, I'd invite my lupus patients to come in and talk with each other. They found it enormously helpful, and I would leave them donuts and coffee and
Yeah.
go around, come back and find out some very interesting things.
Yeah. So that, I think we've talked about the patient group things. What about, there's a couple of things on there relating to the assessments question that, again, this is from the doctor's point of view, I think can be quite a frustration and almost a deterrent to taking part in trials, isn't it? Are there resources that can, just from a practical point of view, that somebody who's going to have to learn to use SLEDI and BILAG or other instruments can use?
Well, there is training, but by and large, my experience is that the individual sponsors take on that responsibility, but that it's often not enough. And so whenever there's online training available, I know lots of investigators will take advantage of it because they don't feel comfortable, and they can't do it in an efficient manner unless they've been sufficiently trained.
I agree. So yeah, they usually, if any of these trials, there's usually something like an investigators meeting where somebody will be asked, maybe me, to explain how to use BILAG and SLEDI. And there's usually that talk there. But if that's the first time you've done it and you haven't done it in a trial, you may need a little bit more, because once you try these things in real life, they become a little bit more difficult. There's also the LFA point system, isn't there, which provides which often sponsors will provide access to.
Yes. Yeah, so those modules I think are very good. Training in SLEDI and BILAG and CLASSI and so forth.
Yeah, they're very important.
But in my own personal experience is that you learn how to do it by doing it, actually, and you have to do these assessments in a real life context, actually. And then how much are the new outcome measures going to help? Is this going to add more complexity, or is it going to make life easier?
I think it's going to make it less complex.
Yeah.
First of all, I think TRM SLE is a very important effort and large scale. And OMRAC always involves patient research partners. So as we're going through the Delphi process to decide on the final core outcome set, patients have been very, very active in that, and it's really quite important. And that should be amenable to whatever new outcome measures are developed, because as we then have the core set of domains, we will look to validate appropriate instruments for each of those domains. And that means that then we can help with validating the new instruments that are being developed.
Yeah. And I actually find the newer instruments have been designed, people thinking about trials, as you said, the BILAG and the SLEDI were designed before it was all about RCTs.
Right.
And I often find if I do something like CLASSI, I often feel like this is really helping me describe what I see.
Yes.
It's
exceedingly important that the measures are developed for trials.
Yeah, and I feel
not for
when I do it,
practice.
it helps me. It's not a hindrance. So we should probably go on to some of the questions now. We've had quite a few questions in the chat, and I'm going to try and sort of group and summarize some of those things together. So some of the questions, maybe I'll go to Rich first for this one. There are several questions about when patients are eligible for trials, we see people in the clinics who are eligible for trials, but they don't always consent. And are there any ways that we can help that to happen? But which could be for any patient, but also particularly maybe for things like ethnic minorities to get into clinical trials.
Wait, so the question is, a patient might fit eligibility criteria, but
how
they're somewhat reluctant?
Exactly. How do we persuade them it's the right thing to do, if it is?
Oh, that's a tough one. So it ranges from trying to be a little bit persuasive to being coercive. Obviously, we don't want to be coercive. So I think you just have to know your patient and just be very transparent.
Yeah.
And I often will have a patient who was in a trial talk with the patient who's thinking about a trial.
Yeah.
I mean, it's scary.
I don't know. Brad or Vibeke, do you want to comment there?
I think I think having patient navigators to sort of do just what Rich said, I've done this, it wasn't so bad, etc., is really pretty critical. And we also, I'm sure you all have too, we have our clinical trials unit. We have very sort of passionate or compassionate nurses that will go through the trial with the patient, and then I always have time to sit there and answer the other questions they have, like, is this drug going to hurt me, that sort of thing. This often comes up. So it's really an individualized approach. I don't have any big ideas on
I mean,
how to
a couple
fix
of
it.
things. A couple of things I think I've learned from inviting people to trial, and people shouldn't be persuaded to a trial if it's not right for them. But a couple of things I've found have been like, firstly, it's quite easy as the doctor to come across a bit like a salesman that you really want them to do, which just makes people suspect what your real motive is, and that's not how it should be. It should be a good match. I've also found PIS sheets are actually almost like a deterrent to do a trial, because sometimes when you read it, it just sounds very litigious and it's full of caveats and we're not taking any responsibility if something goes wrong. It just reads like that, and here's all the side effects. And I just don't find them very helpful, I have to say, and the chat is more helpful. And then another thing, do you find this? And another thing I've found can be helpful to people is, so often people have other options. Like, if you've tried all the therapies that exist and there's nothing else for you except trial treatments, then that's a different thing. But a lot of patients haven't, and they've got numerous other immunosuppressants and things that or licensed biologics that they could try. And actually, sometimes I find if you just lay out the options and leave the trial amongst them, it's actually, people are more receptive to that.
We hear from our patient research partners that some of the major concern is what they don't know. So they're afraid of the adverse events that they may face, whereas right now they're on a therapy and they've come to terms with it. And so when you talk about a new therapy, there's a lot of what ifs, theoretical concerns that have to be at least partially allayed or explained.
Yeah. So I think it's important for the doctor to understand it well enough to explain it with confidence. The safety that's listed in a PIS does not usually match the safety that's actually come out in the phase 2, for example, does it? And I also think from sponsors' point of view, other ways of presenting information, videos, leaflets, things like that could be helpful. So I'll look at some other questions here.
Rich has something to say, Ed.
I was about to say
sorry.
I think it's important to involve and educate the family members. I would say almost always when a patient declines a study, it's not so much the patient declining, it's the family who hears about the study and tells the person in the family not to participate.
And it's also an important one if there's language barriers as well, isn't it? I've found. Another one here, there's also some questions here about other standard therapies for lupus using mycophenolate, cyclophosphamide in different regimens. I think when it comes to trials, I think what's being asked about here is about managing the standard of care. I guess from my point of view, one thing I've often seen is the standard of care is mycophenolate, but people are already on mycophenolate when they develop their lupus nephritis, and then I don't know what to do. Have you got any
Well,
suggestions?
don't you think the standard of care is it's changing now, it's evolving, and I think a lot of it's determined in part by the new therapy and also by what practice has dictated. So even though we consider certain things standard of care, that's not always true internationally. So you have to think about a variety of background therapies.
I agree. Do you have anything to add on that, Brad? Nephritis
Well,
trials?
I I am a little concerned. As Rich has discussed, we have a fixed number of patients that can participate in a trial. And one of the big issues that patients have is, am I going to get a placebo? And as you brought out, if a patient's on a dual therapy with steroid and mycophenolate and they actually flared on that, now maybe if they flared as it was being tapered, that's a different story. But if they flared on that, it's not, in my opinion,
Right.
ethical that you're going to randomize them to get only that. And I just think that now that we have our newer therapies that are approved, at some point we have to sort of bite the bullet, and
Yeah.
those are going to be the background therapy upon which another drug has to do better than, because let's be honest, the two new therapies that are approved provided incremental increases in response rates, right? But they didn't solve the problem of lupus nephritis. So to me, if I were a patient, I'd want to try one of the newer therapies and then plus or minus, the extra thing that's going to be tested. And I think we are going to see a movement toward that direction. The patients are going to dictate it. They're going to say, I'm not doing a placebo trial. I want to be on one of these new therapies first.
Yeah.
We just did a comparison of RA trials over the last 20 years. And it's very interesting because almost all the background therapy for the placebo groups was failed therapy. And, they'd already failed it and they were going to get 3 more months of exposure to a failed therapy. So I agree with you completely. We really should be moving to active comparator studies. And they should be quite feasible.
Yeah.
But there seems to be a fear of doing it.
Yeah, I run a big lupus clinic and I'll do it tomorrow, and I know I'll see lots of active lupus patients, but most of them won't even be eligible for a trial, never mind get the chance to consent to it. So the way these things are designed, I think, could be changed to make them more inclusive. There's a couple of comments in the chat. They're suggestions, really, rather than questions, but I just have to read them out. One is saying, there's a significant number of African ancestry lupus patients in the Caribbean, and we have rheumatologists and patient support groups. This is a population that's ready to collaborate. We have a Caribbean Association for Rheumatology that can be contacted. Pharma are starting to show interest, but more can be done. What do you think? I mean, I think this is generally a very good point, that there are some, in terms of, if you're looking for good patients, there's some untapped resources where there's some great patients, aren't there?
Yeah, I've been talking about that for about 25 years. And in fact, we received a teaching grant about 10 years ago, and we taught at the University of West Indies in Kingston, Jamaica. And at the time, there were 3 rheumatologists, and I suggested that clinical trials be done there, help the patients with resources. But I mentioned that to a bunch of sponsors, and many sponsors said they will not develop drugs in countries or in areas where they will not market the drug. And the other problem, these rheumatologists, the 3 rheumatologists for 3 million people on the island, were swamped. But my idea was to have rotating principal investigators and research coordinators, and I certainly volunteered to go down there in January or February.
It was exactly the same when I spoke in South Africa that yeah, that I was sort of thought this is incredible, the disease that's being seen here and the potential opportunities to make a difference. I guess its sponsors also want the confidence, don't they? The sites are going to be able to do it. You know, they have the resources and the expertise.
So happily, I'm working with a sponsor right now who has asked for connections in Africa. And so what I'm doing is working with someone here in the United States who has good connections, and we're going to see if we can start to bring in some African investigators and open some sites in that continent. And I think these are all, they're just sort of emerging to me anyway, but real opportunities for us to look at a much more diverse population of patients. So I think we seem to be moving slowly in the right direction, but it's pretty slow.
And then the other suggestion actually relates to what you just said, which is, has LFA partnered with Arthritis Foundation to get the word out about trials in underserved communities? Yeah, but outside experience that, but I think there are opportunities there to do it, aren't there, through these different organizations. I know in the UK, Lupus UK are very much focused on this question and what I like about what they're saying is they're thinking it's not just a case of more leaflets and more posters. We've got to think about how we present the information, that it suits the person's not just language, but their understanding of medicine, how they react, how they relate to a healthcare system, things like that. That can all be different, can't they, depending on patient's cultural background. There's about 3 questions. Okay, this is quite technical, but it is actually quite an interesting one, which are all about, can novel urinary biomarkers be used in trials as surrogate endpoints? Would that be better than proteinuria?
I'll take this one.
Yeah, so.
Or I'll start anyway. Yes, actually, not quite yet. But Rich and I are on a committee with the Lupus ABC to start to look at this. And there are biomarkers that really look quite good. And some are even patented by some of the members of the committee that's looking at this, and they're going to be made into clinical tests. I mean, there's still a little bit more validation that needs to be done, but ideally, what we would love to do, to your point, is do we have a biomarker that says, I can look non-invasively at this person's kidney and know that there's no more, with some certainty, with error bars, know that there's no more histologic activity, because that's what you want to do. You want to clean the kidney up and get rid of inflammation. So I would say we're nearly on the cusp of that, and stay tuned, because that will dramatically revise how we do trials, I believe.
I agree. Well, I think we could go on with these questions for a lot longer, but unfortunately, that's all we have time for right now. So thank you to all the panel for speaking and to everyone for attending. At the end, I hope this has been useful, and if you could please fill in a short evaluation form to let us know your feedback. The webinar survey is going to open up automatically once it ends. We really do read that feedback very carefully and think about it for the other resources we're going to offer on Lupus Forum and other webinars that we're going to conduct in the future. So it will be noticed what you've written. Also, as always with Lupus Forum, not everyone has been able to watch this, or you might want to go back and watch some of it again. So there's going to be an on-demand version of this webinar, which will be available on our YouTube channel, and you can also access it from the Lupus Forum website. And of course, if you look through that website, you'll find all sorts of other resources related to lupus. We are thinking about our next webinar it touches on some of the themes that we've discussed today. So look out for that coming soon. If you sign up to our newsletters on the website, or if you follow Lupus Forum, all one word, on Twitter or in LinkedIn, then you'll get notified when that happens. So all that remains now is for me to say thank you to Richard, Vibeke, and Brad. And thank you to everyone for listening. Goodbye.
Bye-bye.